And we are really honored to have Dr. Christopher Gardner today here with us. He doesn't really need introduction. Most of you already know him. He's a renowned professor of medicine in nutrition science. He has more than 25 years of research experience, and he has been the frontier in helping public and scientists figure out how to eat, what to eat, and what to avoid for optimal health. And he has done more than 20 nutrition intervention trials that involved more than 2,000 participants. He's also actively involved with American Heart Association, American Diabetes Association, and Menus of Change. That is a collaborative project involving scientists, business leaders, chefs that focus on unapologetically delicious and healthy food. So we are really honored to have you here, Dr. Gardner, and please, as we said, feel free to raise your hand if you have a question and you want to be unmuted, or feel free to put your questions in the Q&A and I will monitor them, and we can get started. Thank you so much, Dr. Gardner.
Thank you, Susan. I do want to tell everybody how intimidated I was. So Susan invited me to do this, and I said, cool, is it a half an hour lecture or an hour lecture? She said, no, it's 2 hours. And then I got a notice that I was offering a course. I said, a course? I thought you asked me to give me a lecture. And so she explained that 2 hours is a course. And you know, I'm pretty well known for doing about 100 or 150 slides in a half an hour. And she said, 2 hours. I said, oh my God, this is gonna be hundreds of slides. So literally, I have hundreds of slides. But the way I've arranged this is I kind of have 100 slides of how I start the nutrition class that I teach to human biology undergrads. And it's really all about study design. So it's a very nutrition science focused talk today. My class is really about carbohydrates, fats, proteins, vitamins, and minerals. But in the front end, I talk about how we know what we know about with science. And at the back end, when we finish the class, I kind of get into how we apply that to studies. So you're going to get the beginning and the end. The first half, I'm probably not expecting any questions. I'm kind of going to roll through pretty quickly. And then maybe we'll take a bio break at 10 o'clock. And then the last half, I have a whole bunch of my studies, and I have some interactive slides where I'm gonna get you to sort of help me think through designing a study, applying what we learned in the first hour. Okay, now it is, when I share my slides, I can't really see who's asking questions. So I have to every once in a while pop up to see. Don't
worry, I will monitor the question and will let you know.
Actually, that's
you
weird.
focus on your slides and I will interrupt you when the questions come in.
Yeah, actually, I can't even see the chat anymore. I can just see Q&A. Okay, never mind.
That's the thing about, yeah, when you share your slides, it kind of, but you can decide, I think, to go off the full screen.
Yeah.
Yeah, that's the problem with sharing slides. The chat goes on the option, under the options. There is a drop down under the options. I think the chat and everything goes there, but I will monitor it for you. Don't worry.
Yeah, okay. So everybody, Susan's going to help me out here, team effort. Shameless self-promotion. If at the end of this you think you want more, the Center for Health Education helped me in creating an online class. We've actually had 1,000 people take this class globally, and it's been really highly ranked. It costs a lot of money. I don't know if you can apply STAP funds. I'm guessing you are. It costs $2,300. That's not up to me. That was up to the Stanford Center for Health Education. But a little shameless self-promotion if when we're done, you want more. So when I do that online class, it's 8 weeks for 8 to 10 hours a week. And when I teach my human biology undergrads, I get two 2-hour lectures for 10 weeks in a row. For you guys, I get 2 hours. So I'm just going to talk super fast and go through everything in my class by showing tons and tons of slides, maybe. Okay, I'll start with an intro and just say, whew, nutrition is tough. We have all these patterns that we would like people to consider eating. I don't know who's tried keto and who's tried Mediterranean, who's tried vegan, right? And at the end of the day, what we would like is to see if you ate some way, could you prevent the major chronic diseases that we know afflict many Americans and people all over the globe, right? But really, to do that, we see what kind of foods are part of those patterns and what kind of molecules are in all those foods. And on this end, wow, it's really hard to wait for people to get those diseases. So we try to find intermediate proxies, aspects of physiology and metabolism that are risk factors for those diseases. And then, oh my gosh, if you work on this campus, I have so many colleagues that can measure very specific things for all these areas of physiology and metabolism. And I'm sad to say that in my field, one of the reasons I think nutrition is so confusing is to get a study funded and published, you often have to pick one of these molecules and study one of these markers. That's how you get your grant funded and how you run a study and publish it. And then you say, oh yeah, you know, that nutrient came from that food, so it's relevant. And that HOMA-IR, oh my God, that's related to endocrinology. And so that's part of physiology and metabolism. So I think that means if you eat that food as part of that pattern, that molecule we measured is part of metabolism that relates to this disease. And it's actually not that simple. It's really complicated. Plus, I think there's different doses of foods, different food sources of them, and there's different durations, and there's different population issues. Oh my God. Are you studying sick people, healthy people, old people, athletes? So I say my whole field is a data-rich environment, and there seems to be a lot of nutrition confusion, and you are in luck. I'm going to help you resolve all the confusion out there in now less than 2 hours, okay? I'm going to come back to this question later. This isn't a pop-up. It's not a poll. I want to see if you think you could answer which one of these, I think, relevant nutrition questions you could support or refute the most definitively with science. Because the seminar today is about nutrition science. Some questions are easier to answer than others. And an underlying theme for all of today will be answerable versus unanswerable questions. A lot of the questions that I get are super practical. Christopher, is garlic healthy for me? I actually did a $1.4 million study on garlic, and I was only able to show at a specific dose over a specific amount of time for a specific population for LDL cholesterol if it was good or bad. And that's not the answer they wanted. They wanted to know if they should have more garlic or not. And that's not answerable. It has to be really specific. Okay, we'll come back to this question and we'll get into the basics of nutrition study design. This is how I start my nutrition class in HumBio. How do we know what we know? And I'm going to start out with a study framework, and then I'm tediously then going to go through a whole bunch of different types of studies. So you will know the secret sauce behind how we make dietary guidelines by looking at all these different types of science. So I'm going to come back to this slide later and you'll understand it more. But this isn't a clever acronym. It doesn't make any sense. But I think all these factors affect the evidence that we're evaluating. Did you get data from a country or an individual? What kind of nutrition and health outcome data did you get? Was it an observational or an interventional study? Was it cross-sectional or prospective? Did you study a risk factor or a disease outcome? Was it primary prevention or secondary prevention? The thing I like the best is if I have nutrition data from individuals and health outcome data from individuals in an interventional study that was about diseases, not risk factors, and if we were preventing it by doing primary prevention, not secondary prevention. There are almost no studies like that, very few. Oh, by the way, as you know, I'm at Zoom and my kids have to wake up for school. That doesn't start till 10, and I have a bunch of loud dogs. So hopefully my family will help me with my kids waking up for school and the dogs barking when somebody comes to the door. Okay, that's my goal. I want to get you there. And to get there, usually to answer questions, we have to use all the tools that we have: observational epidemiology, human trials when we have them, animal studies, tissues and organs, cell and molecular. I really feel like we rarely have adequate evidence to give really clear nutrition answers. And we draw on all of these. I really want to emphasize, we draw on all of these when you try to make dietary guidelines for folks. And another framework that I like to use is, oh, you see something in the newspaper and it says X is good or bad for Y. And I want you to think, was it a study of a single isolated nutrient or a single isolated food? Or is it more like the Mediterranean or the ketogenic or the vegan diet? Because this is simpler to study because it's isolated. Foods are more complicated, but culture, oh my God, culture is more complicated than food. And then did they study something with an acute onset and an acute resolution, like let's say a cold, vitamin C in colds, or iron and anemia? Or are they studying risk factors like blood pressure or cholesterol? Are they really waiting to see who lived or died of cancer or heart disease or whatever? And so I have found in my field that this is the best place to find the best evidence, but often the least interesting. A single nutrient for a single disease with a quick onset and a quick resolution. You can quickly try different doses and sources and nutrients, and you can do this over and over again until you get the answer. But as you get farther away and to the other corner of this grid, if you want to know if the Mediterranean or the ketogenic diet will prevent diabetes or heart disease, no one's done that, and no one's actually ever going to do that. Can you imagine agreeing to sign up for a study? Say, sure, I'll eat Mediterranean for the rest of my life. Oh no, you changed your mind? Sure, I'll eat carnivore for the rest of my life. Oh no, you want me to be vegan for the rest of my life? No, people aren't willing to do that. And so it's really hard to have chronic disease outcome data for things as practical as should I go vegetarian or vegan or keto or Mediterranean, okay? I think that's what people want to know. What should I eat or what diet pattern? And sadly, that's where it's the hardest to get the evidence. So as I go through my talk today, I'm frequently going to go back to this grid. This is what I do with the undergrads. And then there's the added layer of complexity. Is it an observational study, which is association only, or is it intervention with cause and effect? I'm super proud of SPRC where I work because our focus is behavioral interventions with cause and effect. But they're hard to do and they're expensive and you can't do a lot of them. So I'm going to flip back and forth between observational and interventional and my grid here. And think about this for a sec. Is it possible that nutrient or food X does improve health, doesn't improve health? Is it possible these can both be true? I mean, you wouldn't think so. This says yes and this says no. I actually think this is a problem in my field. Not a problem. This is a tip for you. They can both be true. If you vary the domains of this question, you can make it so it's both true and false. And this is from when I was finishing my PhD, I saw these two papers, 91 and 1990. And one said oat bran does lower LDL and one said it doesn't. And that seemed really annoying for headline, but you didn't have to look very far. This one where it did lower LDL was done in hypercholesterolemic men who had room for improvement. This one where it didn't was done in college-age students who didn't have high cholesterol. They had normal levels. So oat bran wasn't able to lower their cholesterol lower than normal. Can't you have both those things at the same time? Yes, but the headlines made it look crazy. It does and it doesn't. It wouldn't have taken long to look at the study designs and see how they're both true if you can put it in context. So in my field, the term I use the most often is it depends. Give me the context of what it is, and then maybe I can tell you if it's good or bad. Okay? All right, so ready? We're gonna fly through a bunch of designs. I'm probably gonna go a little fast here. I'm gonna guess a bunch of you know this already. Hang on one sec. Can I mute? Jackson, can you wake up the boys? Yeah. Thanks. Okay, here we go. So it often starts with ecological studies because these are inexpensive. You can do these in the library. You basically go say, oh, from one country to the next, what kind of imports and exports do they have? And then instead of trying to figure out what each person eats, I'm just sort of averaging something for that country, as well as averaging rates of disease. I'm going to look at national morbidity and mortality data. I can compare country A to B to C to D. And here's a classic from 93, death rates from coronary heart disease. This is back in the days when we only studied men. Sorry about that. And here were rates of disease in the different countries. And here it was a cholesterol saturated fat index per 1,000 calories per day. And look at that beautiful line. There is a correlation here. If you are in a country that eats more cholesterol and saturated fat, you have higher rates of heart disease. Ooh, not causal, just association. And plus, look at some outliers. Here's Finland and here's France. They actually have just about the same amount of cholesterol and saturated fat. And they have like a 5-fold difference in heart disease rates. So it looks cool, but then there's some flaws with that. So this is one of these observational studies. It's cross-sectional. They're just seeing what's going on in the country that day. They were focused on nutrients in this particular case. And so we've got some associations. So there's a problem with this kind of study and an advantage, right? So for these ecological studies, there's many alternate hypotheses. Oh, there were other things going on in the country that were associated with eating high cholesterol and saturated fat, but that wasn't the cause. So many alternate hypotheses, you can't prove it, but it's pretty quick and inexpensive. You just get a graduate student or an undergrad to go to the library and you mentor them and they look up the data and you get published. So plus in the US, sometimes everybody eats fairly similarly and it's hard to see a broad range of intakes. If you go globally, you can pick some countries with really high versus really low intakes. And so you can actually get sort of a wider spread of things to look at. So there's value here, but not a lot. So we need to go to the next level up for observational studies. I'm going to focus on case control and cohort studies. Okay. So in our case control cross-sectional study, I got an example of tofu. I'm a big tofu eater. And they were looking at tofu risk and breast cancer. Okay, so check this out. They got about 1,000 women without breast cancer, and they asked them how much tofu they ate. These are from LA, San Francisco, and Hawaii. And then they got 600 cases, and they asked them how much tofu they ate. And so here are the rates of, here's how much tofu they ate between the cases and controls. And they used this low intake of tofu as sort of the reference ratio. Can you see how the ratio is changing at the low level, right? There's more cases than controls. And at the high level, if you eat tofu a lot, there's more controls than cases. And if you do an odds ratio and you make this the reference group, you can publish your paper and say, aha, the implication here is that if you were a really high tofu eater, like once a week instead of once a month, you had a 33% lower rate of getting breast cancer. But it's not getting cancer, they already had breast cancer. So this is cross-sectional. It's actually possible that it wasn't eating more tofu that led to less breast cancer. It could be that once you got breast cancer, you changed your diet and you wouldn't know because I don't have a temporal association. Everything's cross-sectional here. So there's a big limit about the cross-sectional nature of this and which came first, the tofu or the breast cancer. But it's interesting. It's sort of a, I ate tofu and I was looking at breast cancer, but observational, association only. I got another one for you that I'm going to bring up later for a clinical trial. Neural tube defects and vitamin use, in particular folic acid, but I'm not really going to get too deep into that right now. They asked about 1,600, 1,700 women about whether they had taken vitamins when they were pregnant. So they had to ask them in the past. And so they found about 600 women who had had a kid with a neural tube defect, which is horrible. And they found controls. And they found another group here that had a birth defect that wasn't a neural tube defect. Now, basically what they found was very few women were taking these vitamins, okay, across the board. These are not different at all. They were really hoping to see that some group was getting 60 or 80% of them were taking them, or at least the controls who had normal births, more of them were taking it to show that taking vitamins is good for you. It's really not there. Why do you think they have this group? I'm going to bring up this term of recall bias. Just think about this. If you're a woman who had a child with a birth defect and somebody said, you know, is there anything that you did that might have caused this? Oh my God, you're racking your brain. You actually might remember differently than you asked a woman who had a normal birth. And so it's possible that some of these women were blaming themselves, thinking, and they actually had taken vitamins and they forgot, oh, I never took vitamins. But they did. They're actually remembering differently, sort of blaming themselves or racking their brains for what happened. So a problem with this kind of study, when you're asking people to remember in the past what they did when something bad has happened, is that they're remembering differently than the controls. This particular study, cross-sectional, would and did conclude doesn't seem to matter if you take the vitamins or not. That's not what's going on. And we even have this recall bias control, a group that had a poor health outcome, but wasn't likely related to taking vitamins or not. Okay, where does that belong? So I'm going to call neural tube defects acute onset because it happens right after the pregnancy. And they were looking at folic acid as an interesting possible nutrient. Okay, remember this one because we're going to come back to it. So a major limitation of case control studies, as with all observational studies, is alternate hypotheses are available. And then that temporal relationship, which came first, the breast cancer or the tofu, and the recall bias I just brought up. The major advantage to case control is it is quick and inexpensive because it's cross-sectional. You can do it all in 1 year, or particularly appropriate if it's a rare disease. I'm going to show you how rare neural tube defects are. I'm going to show you a clinical trial later where they looked at it, but we'll compare the numbers of who they got to study. It's a really rare disease. So sometimes the advantage of case control is that. So keep in mind limitations of the temporal relationship and the recall bias, and the major advantage being rare diseases. So let's go on to the type of study that I think gets more headlines than any other type of nutrition. And I'm green with envy, and so I'm going to make fun of them. It's a bunch of my friends at the School of Public Health at Harvard that do the nurses' health study and the health professionals study. Oh my God, they have a paper in the New England Journal of Medicine every week because this is the game they play. They asked between 3 studies, Nurses Health 1, Nurses Health 2, and the health professional study, which is all men. I think they have a combined 300,000 or 400,000 people that they asked what they'd eaten. And every 2 years, those hundreds of thousands of people get a letter in the mail, say, are you dead or are you alive? Okay, if you're alive, did you have any outcomes that brought you to the hospital? Okay, nothing, you're good, you're healthy. Okay, we're gonna write you again in 2 years. And so over 30 or 40 years now, many of those 400,000 people got sick or died. And so they can just sit there and spin their things once a week. I know it. They sit around the table and say, what do you guys want to study this week? Okay, let's study coffee. Okay, cool. What do you think it should be related to? I don't know. We collected a lot of data. Depression. Cool. So should we study all of them? No. Let's do the men that were 25 to 40. And then somebody says, wait, didn't we do that last year? Oh, you're right. We did it last year. No. That was 5 years ago. Oh, well, if it's 5 years ago, we can do it again because more people got sick or died. And so they often, if you look at their cycle every 5 or 10 years, they redo their analyses because they have more events. Anyway, this drives me nuts. They're really great people and they actually do really good science. But this is the most common thing you'll see in the newspaper in a headline. These hundreds of thousands of people told people what they were eating. And decades later, we can see who got sick and who didn't. And we know what they were eating beforehand. So here's a classic. This happens to be the Iowa Women's Health Study, where they were looking at relative risk of heart disease after 9 years of follow-up. And it had to do with who was eating more whole grains, who ate the most and the least whole grains. So compared to the ones who ate the least, which will be our reference group here, if you ate more and more and more and more servings of whole grains during the week, it turned out fewer and fewer of you had cardiovascular disease after this follow-up. So literally, if you trace this back, this looks like a 30% protection if you ate more. Now, do you have to eat this much? No, actually, there was a 30% protection this whole time. So basically, you just got to get a median of 8 servings a week. 13 and 22.5 don't help anymore. So you basically just got to get over this hump is the implication. But think about this. So what's the alternate hypothesis? What do you think is associated with eating more whole grains that might not mean it's mechanistically the whole grain, it's something else? And I'm pretty sure you can all guess that these folks, if you look, sleep more, exercise more, take more vitamins, eat better in general. And so you can't actually say it was the whole grains. Now, the folks who do these analyses know this, and they actually have sleep data, and they have exercise data, and they have data on their other diet, and they statistically adjust for it as best as they can. But we do have to recognize that the data they have on the sleep and the exercise and the rest of the diet isn't perfect. So they can't perfectly adjust. But when they do this, they do say, independent of other things, this was helpful. And then they have to look for a plausible mechanism. Well, whole grains have a lot of fiber, and I did a rat study, and I looked at their rat's cardiovascular system, and I was able to sacrifice the animal and open up the arteries, and I can see the plaques or not. And so you start building a case for whether this could be real. But this study is just association, not cause and effect, right? So we go to our grid here. It's whole grains. It's a chronic disease, but it's an association. Here's another one. I'll show you some specific numbers in this one. So there's a Mediterranean diet score to see how Mediterranean you are. And you basically, this is kind of a weird way to score it. They find the range of all these things in a diet, in a population. And if you're above or below the median, you get a point. And if you're the other way, you don't get a point. So in this particular scoring, you got a point if you ate more than half the group of vegetables, legumes, fruits and nuts, cereals, fish, and you got a point taken away, or you only got a point if you ate less than half the group for meats, poultry, and dairy. Alcohol is the funniest one. So you get a point against you if you don't drink, and you get a point if you drink a little bit, and you get a point against you if you drink too much. This is how they make the Mediterranean diet score. It's that simple. And then they had 22,000 Greek adults. They followed them for almost 4 years. 275 of them died. So only 1% died. They don't actually have a lot of data, but they had enough. They published a paper and they said, of the 275, what did they die of? Heart disease, cancer, and there were total deaths. And in their final result, they said, look, there's about a 33% reduction from heart disease, 24% from cancer, and 25 total deaths for every 2-point shift in the 9-point scale. So they're suggesting that if you were a 4 on the scale, if you moved up to a 6, you'd get this protection. What if you moved up 4 points? Well, the implication it would be double that. What if you went from a 1 to a 9? Oh my God, you'd be disease-free and you'd live forever. Okay, but this is all based on 275 deaths for 4 years of follow-up in 22,000 Greeks. It's not perfect. Did the people who ate more Mediterranean sleep more? Yes. Exercise more? Yes. Eat a better diet overall? Yes. Interestingly, of all those components I showed you, they did try to look one component at a time to see if that offered protection. And none of them individually predicted health protection or health promotion. It was the sum total. It was being Mediterranean. You can't just cheat and just drink olive oil at night. It doesn't work. You can't have a Big Mac and a Whopper and chicken McNuggets and then drink olive oil at night and say you're Mediterranean. You're supposed to do all 9 of the things. Okay, back to this one later too. Where does that one go? Well, I think of the Mediterranean diet as a food pattern or sort of a cultural thing. And they saw who lived or died, chronic disease, but it's still association. We still don't know for sure what did it, right? So in a cohort study, the major limitations are, again, alternate hypotheses, because it's only observational, it's association. Oh my gosh, those are expensive, long studies. If you're going to track 22,000 people for 4 years, keep the retention up, continue to contact them and get follow-up data from, very expensive. The big advantage this has over the observational cross-sectional studies is the temporal sequences there, because you know what they were eating in the beginning, and then they got sick later. Another thing that I think is practical and advantage of this is if you're doing these cohort studies, you're not asking anybody to change their diet. You're just observing what they're eating And so it's always an achievable range. You always get to compare the people who are the lowest versus the highest. And you know that real people eat that because it was just real people eating. So another advantage I like in these cohort studies is that you know it's not ridiculous, it's achievable because it was what real people were eating, okay? And then this is not the kind of research that I do, but a lot of this research happens on our campus and I cite their studies all the time. I just don't do them, so I'm just gonna quickly go over this. Animal and cell studies, super helpful for giving us mechanistic and biological plausibility data. Here's one that I thought was really clever. Okay, this had to do with soy and breast cancer. So I don't know how many of you have heard that soy contains a set of molecules that the family name of them is phytoestrogens. And molecularly, they look quite a bit like mammalian estrogen. And if you are aware, there's good data show that women who are postmenopausal, who are taking hormone replacement therapy and estrogen for the sake of reducing menopausal symptoms, they've tied that to increased risks of breast cancer. And then they looked at Asian populations and they said, wow, Asians have less breast cancer than Americans. I wonder what that is. Oh, that's interesting. They eat a lot of soy. I wonder if the soy is protecting them from breast cancer, but that seems weird because hormone replacement therapy and estrogen, mammalian estrogen, is a risk for breast cancer. Is there some way the plant estrogen is protective and the mammalian estrogen is harmful for breast cancer? How would you do that in women? Oh my God, that would be a really long study. Okay, so they did it in mice. So they took mouse pups and they injected them with a carcinogen, and sure enough, they got mammary tumors. And then they did this, they took the mouse pups, injected the carcinogen, gave them a bunch of soy phytoestrogen, and they got tumors anyway, and it didn't change. And then they said, okay, let's try this. Let's give them the soy phytoestrogen before we inject the tumors. And in fact, there were fewer tumors when they did it that way. And then they did a last one. They said, wait, let's give them the soy phytoestrogen before we inject the tumors and after we inject the carcinogen, sorry, not the tumors, inserting the carcinogen. And they got the fewest tumors of all. And I was fascinated to hear the guy explain this when he presented these data. He said, this actually looks like what we see when we look at Asian women who migrate to the US and give up their diet and they adopt a more American diet and they have less soy. And so that would be, they had it as a kid and they continued in adulthood versus giving it up. So here's having it in an Asian country, giving it up. And even though you have fewer tumors for being exposed there, you have more tumors relative to Asian women who ate it all the time. And then if you look at American women, you see that they have more tumors. And so they had this very elegant discussion like this. And they said, okay, here's the American women who didn't grow up with it. They hear about it. They try to have it later and it's not enough and it doesn't have any effect. What's going on here? And the whole hypothesis was that in the development of mammary glands, as you're an adolescent, if you're exposed to soy phytoestrogen, there is some benefit conferred and you don't get it unless you're exposed early. So if you do it later, only later, it doesn't work. But then if you actually stay in an Asian country and you get exposed and you get this protection early on and have it later, you get an extra protection. I hope that story made sense. I loved this story when I heard it the first time and I thought, yeah, right, there's no way you can do this in women. You cannot inject women with carcinogens and you cannot play with them like this. And if you can imagine with mice, this can be done in years. In women, this would take decades to do this kind of thing, to see who did and didn't get breast cancer, right? So a summary of what I just said is right here. And I was really impressed to say that they had created this mouse model that seemed to parallel what happened with breast cancer rates in Japan and the US, and Japanese women who migrated to the US versus staying there, including Japanese women who migrated to the US and kept up their intake of soy. It was consistent with different rates of disease in those populations. And so mechanistically, they were studying that in a mouse model, which I appreciated. Now I'm going to go to a bizarre trial that they did in humans about vitamin E. I actually participated in one of these in my doctoral work at Berkeley, where they were trying to see if the LDL particles that carry cholesterol get oxidized or not, and if vitamin E would help this. So they randomized 8 groups of 8 people, and let's see, 8 per group. No. Okay, so maybe 6 groups. 6 groups of 8 people. And this group got no vitamin E, and this group got 60 units, 200 units, 400, 800, and 1200. And after doing this for 8 weeks, they took blood samples, and then they put the blood samples in a cuvette, and they added iron. And what they wanted to do was track over time to see basically if the particles would rust, to see if there was oxidative degradation when exposing the blood and the low density lipoprotein particles to iron. And if there was oxidative degradation, the fatty acids would break down, and you'd have this colorimetric response in a spectrophotometer. And you could see how long it took for the things to break down and the color to change. And the goal was they wouldn't break down. They'd have so much vitamin E in them that they would be resistant to getting this oxidative stress, this iron in the cuvette. And so they were looking for a change in the lag phase of being exposed to this. So all the white was baseline and all the red was after getting the vitamin E. So no difference at low doses. At higher doses, the lag phase increased. So it was longer before the particles were oxidizing and the color was changing in the spectrophotometer. And so they said, aha, this is what's happening. If we give them vitamin E, we can tell from this cuvette after we insert iron in it, which is totally non-physiological, that the protection from this suggests that vitamin E is an antioxidant and it will protect you from heart disease. And this isn't heart disease. This is 8 weeks of getting vitamin E. And then non-physiologically using this method to see if it oxidizes. It makes sense. It's plausible. It's not very physiological, but it's plausible. And so this is where we took a nutrient like vitamin E and we just studied a risk factor. And it looks like we improved the risk factor. Doesn't mean we help disease. We only know about the risk factor at this rate. So when we do animal and cell studies, we have a major limitation extrapolating to human health. It often doesn't extrapolate, but it really helped us explaining some biological plausibility and understanding mechanisms better. Okay, so I tried to go everything that leads up to the gold standard doing the randomized controlled trials. So this is, I whipped you through these. Sorry if that was too fast. Let's see how far, oh, pretty much on time, okay. Okay, what we really want to get to is the clinical trial so we can see cause and effect. So before NIH will give me the big bucks to do the clinical trial, I often have to cite all these preliminary things, particularly all these observational studies. So they don't answer the question, but they're really helpful in making the case, especially if you have some mechanistic plausibility to go with it, that we should eat this to prevent that in a clinical trial. Now I want to intervene on people. I want to do behavioral interventions and I want to track people to see who gets sick or not. And so here is the secret sauce to my success. I'm giving it away for free right now. The double-blind, placebo-controlled, randomized clinical trial. If we feed X versus something else to a group of them at a certain dose for a duration of this, there will be more or less of it. That's it. That is the secret to my success as a professor at Stanford University in the Department of Medicine, the Stanford Prevention Research Center. All you have to do is fill in the blanks, but you have to be really good at filling in the blanks. And I can tell you lots of times I'm not really good because I get so many grant rejections. I get way more grant rejections than grant acceptances. So sometimes I can't even fill this formula out enough to convince reviewers that I have a good idea. So let's go back to vitamin E, which I just showed you. There was a risk factor that worked. Well, here's an intervention. They actually chose this dose partially based on that last study that I showed you. They got almost 10,000 women and men who are sick already. Why do they get people who are sick already? Because they're more likely to die first, okay? And so, sorry, that's kind of morbid, but that's the way it works. So we get people who are at high risk, thinking that we need some people to die and some people to live to have statistics to run to see if we made a difference. We're going to follow them for 4.5 years and see if anybody has a myocardial infarction, stroke, or cardiovascular death, or just the MI, or just the stroke, or just the death, or the total death. Can you see the amazing effect that vitamin E had in this study? It had absolutely no effect at all. None. I think they were hoping it would work, but it didn't. I think this is like health outcomes preventing with vitamin E study. Okay, well, their acronym didn't work. They hoped it would work, but totally didn't work. So it was this one. Oh, this was cause and effect. This is vitamin E and a chronic disease. And if you went back 15, 20 years at this point and asked how many cardiovascular medicine folks were taking vitamin E supplements, almost all of them were. If you ask today, none of them are. Not only did this trial have null findings, but a lot of NIH funding trials got funded at the time because of all the other plausible mechanism data and because of all the observational epidemiology, which I'm going to spare you, and it didn't work. Here was an alpha tocopherol beta carotene study. The alpha tocopherol, that's vitamin E, it didn't work. I'm not going to show you that one for now. They picked smoking Finnish men. At the time they started this study, the highest rate of heart disease was in Finland. And then it was even higher in smokers. Okay, and they had 5 to 8 years of follow-up. And they were trying to see if beta carotene would help. And it was a supplement study. So you got placebo or active agent beta carotene, and they thought it would help with lung cancer. And when they did this study, they actually got statistically higher lung cancer and higher heart disease on the folks getting beta carotene. This is super famous as a horrific result in nutrition. They were trying to save people and actually killed people doing this. Part of the reason is this beta carotene in a supplement form was much higher than you would get from food. How did they pick the dose? Actually, we're going to do that in some of the follow-up. I'm going to challenge you to pick doses. What would be the appropriate dose? What's too little? What's too much? So in this paper, they actually hurt people with this, and they proved they hurt them. It's a randomized controlled trial, right? And so this is intervention with beta carotene. Vitamin E didn't work at all, but it didn't hurt anybody. And the beta carotene actually hurt people. Okay, interestingly, someone went back and said, God, that's so bizarre. Because, you know, when we look at all our observational epi, the people who have the most beta carotene in their blood have less heart disease and less lung cancer. God, how can I assimilate those two things? Wait a sec, I want to go back to this study and I want to look just at the placebo group, because if you only look at the placebo group, it's almost like it's an observational cohort. And so I said, okay, let's look at blood levels in the placebo group. Who had the highest blood level of beta carotene and who had the lowest? And when they did it, the folks with the highest beta carotene in their blood at baseline had the lowest rates of cancer and heart disease. Okay, help me assimilate this. If you have high levels in your blood at baseline, you have low rates of disease later. But if you give people the beta carotene, it hurts them and kills them. How can that possibly work? Why does that even make sense? Well, picture for this placebo group. How did they get high beta carotene levels in their blood? I'm going to oversimplify and tell you they ate a lot of carrots. A lot of beta carotene in carrots. Okay, but eating a lot of carrots means you also got lots of other things with it. Maybe it wasn't actually mechanistically the beta carotene, or maybe it was a lower dose of beta carotene in conjunction with the other things that came in the beta carotene in the carrots. And what they did here is say, you know, we think there's a plausibility that it's just the beta carotene. And it's too hard. Oh my gosh, a lot of trouble to get 30,000 smoking Finnish men to change their diet. Let's just give them a pill or a placebo like a drug. And let's make sure it's a pretty high dose just so we don't miss anything. It actually ended up hurting people in this trial. And it actually has happened in a couple of other trials. So we know that high doses of supplements can have adverse events. And that one, you know, the retake on this was more of an observational study when they only looked at the placebo alone. Here's another trial. Let's go back to folic acid. Remember that one about the neural tube defects that I mentioned? Remember I showed you there were almost 600 mothers with kids with a neural tube defect or another birth defect or control. There's 570 people roughly, women, kids in each group. Okay, so if you really wanted to know, you shouldn't do a case cross-sectional study. You should do a trial. So you got 18,000 women who are planning to become pregnant, randomized to getting vitamins, including folic acid or not. Sorry, I don't have time to go into detail on that, but it's probably the folic acid for mechanistic reasons. And they got a sort of a fake thing, got a trace element supplement and vitamin C. That was the comparison. And then they looked to see how many neural tube defects there were. Of the 18,000 women, only 4,000 got pregnant, because these were planning to get pregnant. This is who got pregnant. And among those who got the placebo or comparison group, there were 6 cases of neural tube defects and none in this one. They totally prevented it here. This is really compelling, randomized, controlled trial. Wow, this is pretty good, right? So we've got an isolated nutrient and we only had to wait a few years to get this. We proved it. Folic acid supplementation prevents neural tube defects. Okay, but think about how rare this is. So there were only 2,000 people in each group. So 1,994 women didn't notice the difference. Only 6 noticed the difference of taking this thing through their pregnancy. Or another way to look at it is they randomized 18,000 women. So really there were 9,000 randomized to each group, and it was only worth looking at the women who got pregnant. Wow, that is a really small number. You have to treat 9,000 women to get 6 benefits. Ah, look at this. Here's an advantage of doing this. Neural tube defects are really rare. Here's an advantage. They had almost 600 neural tube defects. There's a lot more data to work with here. But the trial design is not as rigorous. This is observational. The other one was experimental trial. It's cause and effect. And in fact, we now fortify the US food supply with folic acid. And since doing that, neural tube defects have gone down in the US. I had to put the whole thing together. And at the end of the day, the challenge here, right, is that we all eat a lot of food. And to do studies like this, we tend to focus either on one food, like tofu, or one nutrient, like phytoestrogen in the soy. And then I'd love to focus on all of your health, but we typically focus on something very specific like cholesterol or blood pressure. Most of my studies have to do with risk factors. I don't really wait for people to get heart disease or cancer. I usually wait for their blood pressure or cholesterol or weight to go down. That can be done pretty quickly. Good biological plausibility that lowering the risk factor would lower the disease, but then we only focus on one disease at a time. Otherwise, it's a fishing expedition. And we know that the whole composite of health is very complex. And at the end of the day, what we get are some of these reductionist, isolationist approaches. I'm not going to show you today, but there's a cool thing called a stanol ester that's in some plants. It's kind of like mammalian cholesterol, but it's plant cholesterol. And if you take it, you'll lower your blood cholesterol. And then we know that there's some statins that do a great job of lowering blood cholesterol and saving people from heart disease. And then they make the leap and say, yeah, you should take this new medical margarine that we make and sell that has this in it because it lowers blood cholesterol. And we know lowering cholesterol saves people from heart disease. And that's actually a big leap. You don't know. You didn't actually test this to see if it prevented heart disease. You put it into your model. So I studied isoflavones, these phytoestrogens in soy, for about a decade, and I quit eventually. It was too complicated. I looked to see if they would lower blood cholesterol, and they did a little. I did that because hopefully that would help prevent heart disease. But the whole breast cancer thing came up that I wasn't studying. I might be hurting them. We knew it could be hurting them because women taking hormone replacement therapy that had estrogen for menopausal symptoms had more breast cancer. I don't often get to look at this whole thing. So even when you see that something in food provides a benefit that may lead to prevention of disease, there might be another angle here. I know a lot of OBGYNs have been frustrated. Oh my God, what do I tell these women about soy, which is supposed to be good for the heart because it lowers cholesterol, when it might hurt them? We actually have pretty good evidence that it does not. OBGYNs, you can tell women to have soy. Part of it is that mouse model I showed you before. It probably helps. It probably doesn't do anything for breast cancer unless you are exposed at an early age. And a lot of women in the US weren't exposed at an early age. Anyway, it should be protective, but we don't know. And it's this whole constellation of things going on. And that's why we need to appreciate the different kinds of trials that are yielding the information that we get. Okay. So clinical trials, randomized clinical trials, super important. We can establish cause and effect, and the human relevance is obvious because we do them in humans, and the endpoints are clinically relevant. But I do this for a living. I do randomized trials. I tend to do risk factors, not disease outcomes. And I know that the generalizability of my trials is often really limited because I pick one specific population, one specific dose, one duration. And if people are eating one thing, it also means they're not eating something else. So was the effect the thing that I gave them to eat, or was it the thing that they stopped eating? And there might have been a lot of different things they stopped eating. I tend to study risk factors when I have some classic examples like the vitamin E, where the risk factor, there's a benefit, but you don't change the health outcome. That's happened multiple times in nutrition. And we're usually focused on one health outcome, not overall health, just because NIH won't let us do fishing expeditions. And then we really need to reproduce our results, and they're often not reproducible. At the end of the day, yes, I'm a firm believer in randomized controlled trials. But in my world of nutrition, you get one specific answer to one specific question. And then really quickly, there's so many meta-analyses out there. I just want to mention from a scientific basis, I'm no fan of meta-analysis. People meta-analyze and pool the data from observational studies, interventional studies, both. I see a lot of these. The potential value is it's better than a review. It's actually a statistical approach. You can get a quantitative finding. You can say, here's a p-value. It's not just my opinion as an expert. It's better than that. It's more objective, except it's actually way more subjective than people would like to believe, because you need inclusion exclusion for which trials you will accept for the meta-analysis. If you're really broad and you accept them all, what happens is you get sort of garbage in, garbage out. You end up including a bunch of crappy trials. As soon as you start excluding some, saying, I'm an expert in this field, I know that this trial shouldn't be included, and I know that this study shouldn't be included. Now you're bringing subjectivity back into the whole thing. You're saying, wait, I'll include everything. No, that includes crappy trials. Okay, I'll only include these. Well, then it's not as objective anymore. Then we're counting on you as an expert to have made the right inclusion exclusion criteria. And I can tell you people disagree all the time on what those inclusion exclusion criteria would be. So at the end of the day, when I write grants, when I write papers, I'm always pulling from all these different types of studies. How do we know what we know? It's really rare that we have adequate evidence to definitively say, eat this and that will happen. But to do it, we pool all these kind of things that I tried to summarize. And that's why I would love to have individual nutrition data, individual health outcome data. I'd love to have an intervention that waited for a disease outcome and started early enough that I prevented anybody from ever getting sick, but I hardly ever get to do this. And in my field, it's almost never done. So hopefully this slide makes a little more sense now after everything that I just went through. This is what I look at when I'm trying to answer my questions. And so let me go back to this slide and give you another minute. I'm not quite going to make it interactive yet, but I want you to look at this again and see if you feel any more clearly about which of these could be most definitive. Can you tell? What do you think? Okay, I'm just gonna, let's see. Oh, I can see.
Answer comes in 1D.
Okay. NIH doesn't let us fish. Does it mean we're doomed?
C,
We need private funding.
We have A and C, and then C again.
Okay, so I'll tell you what the answer is. You ready? The answer is, we'd have to look up, I haven't looked at this for a long time. The top one. So this is a nutrient and an acute onset of something. So I can vary the dose and the source, and everybody gets a cold. So I can do multiple trials until I nail this. And I can tell you, this is really sad. We've done a bunch of trials, everybody, and vitamin C doesn't prevent colds or shorten their severity, and people take them anyway. Go ahead and get back to me on another day. How many of you have prevented all your colds by taking vitamin C? It doesn't work, and we actually can do those studies. It's an answerable question, and people still take it like it's a religion or something. Okay, I tell you why this one is hard for two reasons. One, breast cancer is a really hard thing to study, definitively. You have to do randomized trials and wait decades to see who does or doesn't get breast cancer. And a vegan diet, some people have a crappy vegan diet, some people have a good vegan diet. So this is actually hard to define the diet and a long term to wait. Calcium is good. Oh my gosh, it's a nutrient. I can vary the dose. Hip fractures, harder to study than colds. You gotta wait till women, and mostly women, sorry, are in their 70s, 80s, and 90s to get hip fractures. This is hard to study. And then I do this for a living. I study low fat versus low carb. These are really hard to define. What is a low fat diet? I can think of a whole bunch of different options. And what is low carb? Is it Zone? Is it Atkins? Is it keto? Is it South Beach? So this has a huge problem in definition. This is the question you can answer most definitively because it's an isolated nutrient and an acute onset. All of these are more challenging with the scientific tools we have in terms of defining the vegan diet or the low fat or low carb diet, waiting for the breast cancer or the hip fracture. Okay, does that make sense? So this is my world. I'm trying to come up with answerable questions. And sometimes the most answerable questions are not as interesting as the ones that are unanswerable. Christopher, I don't care. I want to know if I should take calcium for my hip fractures. That's a much more practical question that's so much harder to study and have a definitive answer for. I have a whole lecture on this in my nutrition class. It's fascinating. Yeah, it helps if you take it, but it doesn't work as a public health measure because most women don't take their calcium regularly. So it actually works if you do it, but most people don't. So it's not a good public health approach because we don't have good adherence to it. Okay.
There's a question here. It says,
yeah.
Since NIH doesn't let us fish, does it mean that we are doomed or and have to get private funding, or are there other ways?
Yeah, so I would be a perfect example of that. I would say in my 20 years, half of my funding is NIH and half is private. Because I'm trying to study the microbiome right now. And Justin Sonnenberg and I submitted 4 R01s and they all got rejected. And we've now done 6 studies with private funding. And we have another NIH grant in. And I'm hoping this time when they've seen how much we've done with it, that they'll fund our R01, which is going to be reviewed on February 17th. So I'm on the edge of my seat. It is frustrating. Getting funding for nutrition studies is really frustrating. And I'm super happy to be at Stanford with the support I get because they've enabled me to get a lot of funding and do a lot of studies, some of which I'm going to share right now. Okay, so hold these thoughts because we actually...
There's another question, if I may.
Yeah, sure.
Absorption of the nutrients by the intestine may play a role. How nutrients are utilized? Is there any study that correlates this hypothesis?
Yes, you definitely have to absorb it to be able to use it. And so I wouldn't have time to go into that in any detail. Is there any study that correlates this hypothesis? There's things that can increase absorption and decrease absorption, and you'd have to go nutrient by nutrient, population by population, and outcome by outcome. Okay. I'm going to go back to slides. I'm going to give you guys a bio break in just a sec. My next section is really short, so let me do this next section. And then we'll see if we want a bio break, or I'll answer some questions while some people take a bio break. Okay. Okay, so think about this. How many of you drink coffee? I bet a lot of you. Is it good for you? Is it bad for you? Isn't that a simple question? Why don't the scientists study this? Why don't they decide if coffee is good or bad for me? I've heard both things. Okay, it's just a food, right? Can't we do an intervention study with coffee and see if I'll live or die or if something good or bad will happen? Okay. Coffee is a fantastic example. When they used to do this with observational epidemiology, they found that people who drank the most coffee had the most lung cancer. Oh shit, that was the initial observation that maybe coffee is bad for you. Turned out when they did this decades ago, most people who drank coffee smoked a lot. And if you separated out the people who drank coffee and didn't smoke, the people who drank coffee and did smoke, it turned out that lung cancer was totally due to the smoking, not the coffee. Coffee was actually good for you. Okay, so this is why maybe we needed a trial, or this is why in the observational studies, they can try to adjust for confounders, and they did. This is a classic example in the world of nutrition and confounders. Okay, so I want you guys to help me design a study. The NIH just gave me $4 million, you and me, and we get to do it. We're gonna see if coffee is good or bad for you, except, oh, now that they give us the money, we have to tell them exactly. So what dose do you think we should use? And you know, you have to have group A and group B. So we can't have everybody drinking different amounts of coffee. We have to standard, can everybody agree what dose? I bet you can't. Okay, now, do we have to make everybody drink black coffee? Is that fair? What about the people who like mocha frappuccinos? Does that even count as coffee? Should we count mocha frappuccinos and black coffee? Is it like any coffee? How about what about the latte and the milk and the sugar you feel? Oh crap. There's sugar and there's dairy and there's, oh, wow, coffee isn't as simple as just drinking or not, right? And should we include everyone? Or should we focus? Young adults, no, men with cancer. Are we gonna measure cholesterol? I can measure cholesterol in a couple of weeks, but what if that doesn't change your heart disease? Maybe we should wait for heart disease or cancer. Okay, but that will take decades. Okay, we're not doing that with just $4 million. What if we studied joy? Isn't there something to joy here? What if we studied coffee and joy? Do you think the NIH would fund that? I bet they would not, but isn't it important? And should we fund it should we have them drink A versus B for a month or a year or a decade? You know, for some of these, we need 100,000 people for a decade. And for joy, you know, I think we could do that with 100 people in a month. So if you're wondering why we haven't been able to tell you if coffee is good or bad for you, you'd have to think how many people would be willing to say, yes, I'll sign up for a randomized trial and I promise I'll drink 4 cups of coffee every day for 10 years, or I don't mind, I'll have no coffee a day for 10 years. Who would sign up for that? Right? I could say people, you know, I drink coffee, I'd be happy to drink a little more. How about drinking none? Oh no, don't take away my coffee for a decade. I can't do that. Or the people who don't drink any, yeah, I don't mind. I won't drink any coffee. Yeah, but what if we randomize you to the group that drinks 4 cups a day? Oh my God, I'll never sleep, right? Okay, how about alcohol? Oh, don't we know if we should drink or not? Do you know the new dietary guidelines said men should drink less? They did. They used to say women could have 1 drink a day and guys could have 2. And the new dietary guidelines say, no, nobody should have more than 1. Okay. How did they base that? Is that based on a new randomized trial? No, there's no new randomized trial. They looked at the availability of data in a bunch of places. Now, could we get a trial funded so we would really know? Couldn't we just say, okay, drink or not drink and see if you live or die? It's possible. Okay, but is it, and we actually have observational data that suggests alcohol is protective. If you look at the observational data, it says it's protective. But the dietary guidelines just said to drink less because they don't think so anymore. If you look at the totality of evidence, I want to tell you that just recently, the NIH funded a massive $100 million trial of alcohol. But does anybody remember in 2018, this became a huge debacle, that the money actually had come from the alcohol industry to fund the trial. The whole trial was halted. Some of my friends were actually sites for recruiting the people, and they had started recruiting people who willingly agreed to be randomly assigned to drink regularly or to be abstainers regularly. I think that would be a bitch to recruit for, to be perfectly honest. But they did, they'd started it, and the trial got halted because of the politics of who provided the money and the conflict of interest. So that is unanswerable. No one's ever going to do this study, but we still have dietary guidelines. How did we get them? They put together the totality of evidence of all the types of things I showed you. Is it perfect? Nope. Really should do a randomized controlled trial. Could we double blind it? Yeah, right. You're going to double blind people. I don't know if I'm drinking alcohol or not. Something's going down my throat. It's kind of bubbly and fizzy and I'm drunk now, but it might have been the placebo drink. No, you can't blind. I get these ridiculous comments sometimes. How come you didn't double blind it? It's food. You can't double blind food or coffee or alcohol. Would that strengthen the design? Yes, it would strengthen the design. Is it possible? No, it's impossible. Okay, so in all these studies, even when you get to trials, there's a range of stuff around rigor and generalizability. And the kind of trials I do, you can imprison people, you can incarcerate them, or as Kevin Hall likes to say, domicile them, and you can feed them all the food. So you have total control over what they're eating, and you know they're not eating anything else. In my kind of trial, I recruit people and give them advice, and I try to get them to change what they're eating. And so Kevin Hall has done these amazing studies with huge rigor, with sample sizes of 20 for 14 days. And a couple of years ago, we published a big weight loss study of low carb and low fat that I'm going to come back to. 600 people for 12 months. Did I really get people to eat low carb and low fat? Wow, their adherence was actually all over the map. But I think we had the advantage of generalizability. And when Kevin does his studies, he has the advantage of rigor. And so some of the people said, oh, you should have done this. And some people criticized Kevin and said, oh, you should have done that. In my world, we need both. I need small, highly rigorous studies, and I like to have bigger generalizable studies, but I also like to think that we can raise the bar. Kevin does a really good job of making his trials as generalizable as possible in his high rigor situation. And I try to make my generalizable trials as rigorous as possible, taking advantage of all the fabulous Stanford folks that we have to help me collaborate on things and trying to raise the bar all together. Okay, Susan, let's pause for a couple of questions. I actually am going to run through some studies that we have done. And I kind of have some exercises to make you think, wow, what were they thinking when they designed it that way? So the last 50 minutes is supposed to be interactive, but I'm going to pause for just a minute. So why don't you take a bio break or ask a question for
We have already 2 questions here. If anybody wants to take a break, by all means. But we have 2 questions we can answer right now under the Q&A.
Okay, and I got them. I can read them, Susan. So in a nutshell, can you please give us a summary? What diet, as well as how much often it can help prevent and reduce risk of cardiovascular diseases? No, I can't tell you that in a nutshell. That's an unanswerable question. That is perfectly practical. And there's no way to do it. There is no way. You can look at the dietary guidelines. And if that's actually going to be my last slide, is sort of an aspirational diet. And I'm going to say it's a whole food plant-based diet, which means it's mostly whole foods and it's mostly plants. Does that mean you can have some meat and eggs and dairy? Yes, absolutely. Would you do better without any meat, eggs, and dairy? If it was a whole food plant-based diet, you wouldn't do much better by going from mostly plants and whole foods to all plants and whole foods. It would be trivial. It's really, the problem we have in the US is too much added sugar and too much refined grain. So the whole food thing addresses that. And then the plant-based diet doesn't mean vegan or vegetarian. It just means you got lots of veggies and beans and fruits. And so that's how you should reduce your cardiovascular risk. Okay, so I answered it live. What percentage of diagnosis type 1 and type 2 is related to nutrition versus genetics versus lifestyle versus other? Fabulous question. I know plenty of people who eat well and do all the right things and have great families. And I have a good neighbor down the street. Their kid got diagnosed with type 1 diabetes just the other day. And so that is a lot of genetics. And I'm not sure how much, environmental exposure it is. Type 2 is very largely nutrition and lifestyle. We could totally get rid of type 2 if we could manage weight, eat a whole food plant-based diet, get lots of fiber, check out our microbiome. So type 2, definitely. Okay, I'm gonna move to the third one. Oh, might be a first world problem. What about for parents who are wondering, does prenatal consumption of food make my kids smarter? Are these trials realistic? Nope. Easily funded? Nope. Because smart is not a risk factor for disease. Now, smartness and intelligence quotient, there's I don't want to get in trouble here. I'm sure there's genetics involved there. Is there nutrition? You know, in nutrition, it tends to be the case that once you have adequate nutrition, and in the first world, whoever wrote this as a first world problem, in the US, we really do have adequate, we have access to good variety and a largely adequate diet. There's certainly some problems with it, but if you get, there's a difference between getting enough nutrients and more than enough. In nutrition, more than enough doesn't usually count. If you find somebody with deficiency and you address it, you see a huge benefit, but it's really hard to hedge your bets and say, I'm getting a great diet, and so I want to get more. More doesn't often help. It's more sort of like more of that beta carotene hurt in that trial instead of help. Okay, I'm only going to take one more, and then I want you to participate interactively with me, okay? All right, among others, revisiting keto-style diets for brittle diabetics. Ooh, Fred Hex group, successfully getting diabetics off medication, lowering their A1Cs, while also surprisingly to me, changing their lipid profiles. Where does the evidence start? Oh, my last example is keto. We just finished a keto diet and we're writing it up. So I want you to walk through that with me, okay? All right, bio break over. Here we go again. Hope you had a chance to use the restroom, or I hope you enjoyed some of those frantically answered questions. Okay, let's try applying this. Are you ready? Again, we're going to go back to this. And I had to deal with this just in the fall of 2019. In the Annals of Internal Medicine, a report came out, a clinical guideline. Don't get me started. It might take me the whole hour to do this if you get me started on this, where they concluded that we should continue to eat current amounts of unprocessed red meat, although that's a weak recommendation, and we should similarly continue to eat current amounts of processed meats. Unprocessed and processed, go ahead. The main conclusion here was there's hardly any evidence for this. We're really missing clinical trials, and since we don't have them, go ahead and just eat whatever you're eating. My colleagues and I went nuts when we saw this. This drove us absolutely bonkers. Okay, so try to come up with a clinical trial for red meat. Try to use my formula. If these people eat that at a dose of something instead of something else for this amount of time, there will be more or less of it. Okay? Okay, the NIH just said, oh yes, red meat is controversial. Let's give Stanford and the Gardner lab $1 bazillion this time. And first, they're going to have to give us a protocol. So we'll have to pick a type of red meat. We'll have to pick a dose. For the people who aren't eating red meat, they have to eat something else. And we'll have to see what kind of outcome we're waiting for in a particular population. Okay, sorry, I'm going to choose for you because we just don't have time and the format doesn't allow. I think we should pick steak and hamburgers, and you should either have 2 versus 6 ounces daily. We're not going to have none. We're just going to have a lot less versus 6 ounces daily. Or the people who get 2 have to replace those 4 ounces of calories with beans. And we're going to wait for heart disease, and we're going to use heart attack survivors to make the study a little shorter, because heart attack survivors are more likely to have a second heart attack than the general population is likely to have one. Okay, do you know how long and how much money that study would take to be sure that they were really eating 2 versus 6 ounces? Do you think anybody would cheat and eat less or more? Do you think they'd eat all their beans? This would be massive. This is like, holy crap. This is like a $100 million study, seriously, right here. And I get it, and it's published in JAMA, and I'm feeling really good. And the marketing folks said, yes, Stanford has done it. They have answered the red meat question, yes or no. And the first question I get back is, yeah, well, but what about pork? I actually was thinking about bacon, and I'm more interested in 0 versus 4 ounces. And if they don't eat that, I was kind of interested in how that would compare to poultry. And I have more of a cancer focus, so I would focus on cancer survivors. So what does your study tell us about that? And I say, nothing. I didn't do pork. I didn't do poultry. I used a different dose, and it was heart disease focused. Can I have another $1 bazillion and wait another 10 years to do this study? And then I'll know about red meat and health. Except then, as you can tell, though, somebody will have a different permutation of all those domains, and I won't be able to answer it. So these people were crying out for more trials. These trials aren't going to happen. No one's going to fund that. No one's going to agree to be randomized to eat red meat forever or to not eat it. We have to use the totality of the other evidence. And all these groups, all these health organizations have pretty much said, Americans eat too much red meat, eat less. And they say, oh, but you don't have definitive answers. Well, we're never going to have the randomized controlled trial. And if anybody wants, this is a new slide that's actually not in the deck that Susan has, so maybe I'll have to update it. Pretty funny, this doesn't even have an actual issue number yet. I kind of wrote an editorial on it and it got published over the weekend. And it was sort of to address people who are going to say, oh, there's another observational epidemiological trial on red meat, which is worthless. Isn't it? It's not worthless. We're not going to have a trial. And what they actually did in this observational epi study is did a great job of addressing instead of what. And they used a new twist on their observational epidemiological data to take it farther along the pathway of causal inference. They didn't get all the way to cause and effect, but it was really clever what they did statistically and methodologically. And since we're not going to have a red meat trial, they helped. They added to the database. And the next time I'm involved in one of those groups that's making dietary guidelines, I'm going to pull up this study that dealt with type 2 diabetes. It didn't deal with cancer and it didn't deal with heart disease. I assure you this group is going to publish on cancer next and then on heart disease next. They just keep cranking out papers like this. And then the Twitter world will have a heyday. In fact, I posted this on my Twitter account. Boy, the carnivores are mad at me. They're just accusing me of being biased. And oh my God, what a horrible world, Twitter sometimes. Okay, anyway, that's available. Okay, how about low carb, low fat? This is something I've done 2 big NIH studies with. Okay, here I'm thinking about the nuts. Wait, nuts are pretty healthy. They're high fat. What is this thing about low fat is good, but wait, nuts are, okay. So help me work on this one. Low fat versus low carb. My interest is weight loss, and I think I should do it among adults who have overweight or obesity. And the question is which one is better? Okay, so I've actually given you a bunch of this. I've filled out most of these already, and I hope you agree that's a pretty practical question. Which is better for weight loss among adults with overweight, low fat or low carb? So I've got the if. We take these people. I got what to eat, low fat. I've got what to compare it to, low carb. I'm going to do it for 12 months. That's actually what I got funded to study. I was trying to see if I could lower weight or body mass index. Okay, tell you what's missing here is at a dose of, ooh, what do you guys think the dose should be? It's a pretty standard question, low fat versus low carb, but what does that even mean, dose? Oh, I want you to wrestle. I want you to struggle. How low is low? Are we talking about grams, 20, 50, 100 grams of fat or carb? Are we talking percent of calories? I can assure you, if I submitted a grant and didn't tell you how many grams or percent of calories, it would come back to me and say, nope, not funded. You didn't define it. What do you mean by low carb and low fat? And if we had a more interactive opportunity here, it would be fun to do a polling. But Susan, I thought that would take too long. And I can tell you what the answer would be. You all wouldn't agree. There wouldn't be a consensus over how low is low fat and low carb. So I'm going to tell you how we got this funded. And we'll see if you want to criticize me or not. But it's too late because I already got funded and I already published the paper, so it worked. Okay. We call it, this is just going to roll right off your tongues, the titrate limbo quality method. The titrate was, in the first few weeks, up to 8 weeks of the study, how low can you go? And we gave everybody 20 grams a day as a goal of carbs or fat. Now, I hope you know that people get 300 grams of carbs a day, typically. So this is massive. And they get about 100 grams of fat a day. And this is a massive reduction. And I actually think when you get that low, to eat the foods that you need to eat to get that low, I don't think you get all the nutrients you need. So we told all the participants that we were trying to psychologically anchor them And we weren't sure they should be that low, but we wanted to make them feel like we're in a study, we're supposed to lose weight, we're really supposed to change our diet, we're going to take this really seriously. And I said, once you get there after 8 weeks, and to be honest, most of them did. We had 600 people in the study, 300 on each diet. You could titrate up. If this isn't working socially, if you're hungry, then it won't be something you're able to sustain. So add a few grams of carb or fat back and see how you do. If you're still doing okay and losing weight and you're still hungry, add a few more back. And if you find you've added so much back that weight's coming back on and you really could have done better than that, titrate back down. So find this place where it's the lowest you can be, but it's sustainable and you're not hungry and you think when the study's over, if it really worked for you, you could keep that up. That was our definition of limbo, psychologically anchoring you, and then titrate. And then the other thing we said is, and you can't do this with packages of processed crap. Don't go buy the brownies because they say low carb or low fat. We want you to eat a whole food diet. And so if you're on low fat, we want you to get skim milk and organic whole grain pasta and fresh berries at the market. But you really shouldn't have avocados and a lot of fatty meats. For the low carbers, we said you should get organic, whatever you want, farmer's market, avocados and eggs. And you should get some fatty meats, and not a requirement, but if you can get grass-fed, good quality meats, do that. But you really shouldn't have any skim milk, and you shouldn't have pasta, and you shouldn't have fruits. But everybody should have a salad every day. These actually don't have a lot of calories, but they have a lot of fiber and they have a lot of nutrients in them. So even the low carbers, we're told to have a lot of non-starchy vegetables every day. Basically, everybody got told to have a salad every day. And these folks ate here and these folks ate there. So we called it limbo, titrate, quality. Doesn't that just roll off your tongue? The Gardner method of defining healthy low fat and healthy low carb. And this is what they ate. So here's the graphic from the paper. They reported eating more than 2,000 calories at baseline. And I'll just quickly show you that the protein didn't change much. But look at their, 35 versus 45 for carb and fat. So in the beginning, when they were the most enthusiastic, we had huge differences in carbs and fat. And then by 6 months, they had slipped a little. By 12 months, they slipped a little more. But these are, in my world, these are huge differences. This is 30% fat versus 45. And this is more than 45% fat versus 30% carb. These are huge differences. They really, as a group, ate very differently. And this is with advice. We didn't feed them. We didn't provide the food. We had fabulous dietitians. Okay. Do you agree with dose? Did I pick the right dose? Did I pick the right description? Do you think any of my colleagues said you did it wrong? A ketogenic diet is low carb, and that's 20 grams of carbs a day. And your folks were higher than 20 grams of carbs, so you didn't do low carb. And our response is, we actually told them to get to 20 grams. And if they couldn't sustain it, we let them get higher. And they said this is the lowest they could go and maintain, because people who go off a diet just go back to what they were at before. So this is supposed to be sustainable. So I can tell you that the grant was funded and the paper was funded. And so apparently somebody bought into our explanation that this is how we were going to define low carb and low fat. We even looked at glycemic load, which is a combination of the glycemic index and how many of the carbs you're eating. And we really did get a much lower glycemic load in the healthy low carb than the healthy low fat. So this is supposed to be a big thing, the insulin carbohydrate hypothesis, so the raging thing about low carb is better for weight loss and satiety and things like that. We're pretty proud of how we defined it and the adherence we got in a generalizable study where it wasn't a feeding study for 4 weeks, it was for 12 months. And the result of our study was this huge range of response in both groups. We had in both groups, healthy low carb and healthy low fat. These are kilos. We had people losing 20, 25 kilos. And we had people gaining 5 and 10 kilos on each diet. And those aren't outliers. You can see how this is a total continuum. And I could get into this more about how adherent. These people were more adherent to low fat and more adherent to low carb. We have a postdoc who's done a great job looking at the quality of the diets. If you got higher quality low fat or low carb, you lost more weight. So it was a combination of being as low as you can in low fat and low carb and as high in quality. And if you didn't get very low and you didn't choose quality, that puts you in this category here. So I don't know, did I pick the right, how did I do for dose? You guys think I picked the right dose or the right, I didn't actually define it. I let the study population define it. I let them go as low as they could go. And we gave them a lot of support. And we tried to make it as generalizable as possible and as rigorous as possible. And at the end of the day, the average was identical. We had to conclude that, you know, healthy low carb and healthy low fat are the same. The challenge is getting to a healthy low fat and a healthy low carb diet. And I think there's some other factors involved. My next study may be on satiety. I think there were important differences in satiety here. But there was actually the whole point of this study, if you look at the conclusion here, we actually thought there was a genotype predisposition and a metabolic predisposition. It wasn't really just to compare low fat and low carb. And neither of these worked. We thought maybe you were predisposed to do better on one or the other. And it wasn't. I had a good plausible reason for both of those, and neither of them worked. So at the end of the day, we said, you know what? Both of these are options for you. We encourage you to eat a healthy low fat or a healthy low carb diet, but in personalized nutrition, both of those could be options for you.
Dr. Gardner, you mentioned quality. There is a question actually here.
Yep.
Participants, all the whole foods can be expensive. Does everything
Yes.
have to be organic to be whole?
No, definitely not. So organic isn't even more nutritious. The biggest deal, I teach a whole class on organic stuff. To be honest, the person who benefits the best is the farm workers. Organic has a very, very limited set of pesticides they can use. So the farm workers get less exposure in terms of inhaling it and touching it. There's even arguments about the environment. It's a trickier subject than you think. So organic costs more. That is not, you don't need to buy organic.
There you go.
Sorry, I'm mumbling a little bit there, but no, that's not essential. Quality is more about getting seasonal vegetables. Think about how inexpensive broccoli and cauliflower and cabbages, dried beans, so many different dried beans or grains, whole grains. I make a really great wheat berry salad. Instead of having bread with wheat bread, how about making a wheat berry salad or a 3-bean salad? Some of those can be very inexpensive with some small amounts of herbs, right? Vinegar, oil. Yes, you could get olive oil. You don't have to. You could get a less expensive oil. If it was a whole food plant-based diet, it doesn't have to be expensive. Can it be? Yes, in the greater Palo Alto area, it can be super expensive, but it doesn't have to. And you get most of the benefit just from getting the lentils and the beans and the apples and bananas for fruits, which aren't expensive. You don't have to get mangoes, and having dried beans, dried grains, whole intact grains. How about that for an answer? Keep going.
There is one or two more questions. I don't know if you want to take it or wait for the end.
I'll take one more.
Okay, this one is a follow-up to the previous question they asked about H group. Is for a true keto diet, high fat, but only moderate protein and low carbs. So protein restrictive too.
Okay.
So I don't see the
Wait for that one
question
till the end, because I have a keto thing that I'm going to show you. So
okay.
hold
We'll
off.
save it for the end. Yeah.
Okay, I'm going to give you another thing here. Okay, sugar. Americans eat too much sugar. So I happen to be on the American Heart Association's nutrition committee when they came out with really strong recommendation in 2010 to eat less sugar. And Americans wrote and said, does that mean we should eat artificial sugar? And the American Heart Association, we didn't think about that. We wanted to tell people to eat less sugar. We didn't say you should eat more artificial sugar. So I actually got assigned to a writing group, and we had to look at the data and give a position statement on artificial sugar. You ready? Okay, let me just tell you that the US eats more sugar than anybody in the world. It's pretty scary. And interestingly, we eat a whole lot of high fructose corn syrup, whereas most other countries don't. But if you put them together, High fructose corn syrup is basically just sugar. And so we eat a lot. There's a lot of room to cut back. So let's design a study. Come on, team. Okay, and here is the very generalizable, simple question. Should we eat them? Are artificial sweeteners, which are also known as non-nutritive sweeteners, if you want me to explain, I will, but just let's call them artificial sweeteners for now. Are they a healthy substitute for sugar? Sweetener, what word should we use? Sugar, so let's use sweeteners. And so let's try to frame this question as a testable hypothesis, okay? So what are we gonna call sugar? Well, all these things are sugar. Oh crap, you can't just say sugar. We actually had to say sugars throughout the whole paper because there are so many sugars. And then how many artificial sweeteners are there? Well, you guys are all familiar with these, which are about 200 to 600 times sweeter than sugar. So do you guys know that most of the white stuff in these is filler? There's only a couple milligrams of sucralose and saccharin and aspartame in these. And if you only added enough to be as sweet as sugar, you wouldn't even be able to see it. So to trick you, they have to put a bunch of filler in here. So it looks like you're adding a teaspoon of something, but it's so much more sweet than sugar. It's a tiny, tiny little amount. You probably haven't heard of acesulfame K or neotame, but if you look, you'll see this in a lot of foods and beverages. And now we have stevia, which is the all natural. It's not all natural the way they extract it and put it in a powder and people are using it. It was, if you chew the leaves, it's natural. How many of you have been chewing stevia leaves lately? I bet not many of you. Okay, so how can we, wow. Okay, so there's different populations and you'd be eating something with artificial sugar. Which one? Do we have to compare all? Oh crap, there's a lot of them. Which dose? Oh, there's a lot of different doses. And certainly we're gonna replace sugar, but it's sugars. Oh my God, there's a lot of different sugars. Wow, it's more complicated than I thought. And how long would we study it for what? Should we study it for blood glucose? Should we study it for weight? Okay, I'm gonna skip that for now and just tell you what our writing group did. We found that all of those artificial sweeteners I showed you are approved by the FDA as being safe. Okay, that's not very encouraging. It won't kill you. Wait, I thought I was having it so it helped me. Okay, so let's just get out of the way. In theory, they're safe. Let's not debate this for now. It looked like they could be beneficial and they could contribute to weight loss or weight control if you use them to replace current products with added sugars like a diet soda instead of regular soda, coffee with sucralose instead of a thing of sugar. When it got to foods, it was a lot harder. So if you wanted to have cookies with artificial sweeteners, they're still cookies. They're still refined grain and they still have lots of crap in them. You take the sugar out and you put stevia in them, they're not health food all of a sudden. And the other thing that had to happen when we saw these studies is it would only work with weight loss and glucose control, et cetera, if you didn't compensate for this. Because what we found in a lot of studies that had been done, they were only done for one meal. And if you looked at studies that were done for the rest of the meals in the day, we found that people compensated. Like they had a diet soda for lunch instead of a soda. And when it came to time for dessert at dinner, they patted themselves on the back and said, oh yeah, wait a sec, I had the diet soda for lunch. That means I can have chocolate cake for dinner. And if you looked at the whole day, a bunch of people were either completely or overcompensating, and they're getting more calories and more sugar for the whole day. Plus, we're perpetuating a preference for sweet taste. Plus, the things that have artificial sweeteners are crap. It's never in red bell peppers. Did you see a red bell pepper with stevia in it? Did you see garbanzo beans with acesulfame K? No, it's always in cookies and crap. So a huge challenge was compensation, that people compensated later for having their non-nutritive sweetener then, and that the things that we were getting people to eat instead of things with sugar were still crap. They were still cookies and things. So here it is. I'm the lead author of a scientific statement from the American Heart Association, published in Circulation and Diabetes Care. And I want to show you how compelling our conclusion is here. At this time, there are insufficient data to say what to say. We spent 2 years on this. Our conclusion is that it's unanswerable. We don't have an answer. Oh, are you disappointed in me? I really worked hard at this. I had this whole team of super smart people, and we looked at all the literature. And of course, people wanted us to say yes or no. You should have artificial sugar instead of sugar. And we couldn't. It's an it depends question. It depends what you're having it instead of. It depends how much you're having. It depends on a lot of things. And we didn't have a simple public health statement. We do have one for sugar: eat less. Americans should eat less sugar. They eat too much. Should
There's
you?
a question that is,
Go
oh,
ahead.
sorry.
No,
yeah,
go
the
ahead.
question is saying, how about natural sugars in like raw honey and agave nectar, maple syrup?
They're all the same. They're all just, they don't have nutrients in them. They get absorbed really fast in your bloodstream, and they cause a glucose spike, and that causes an insulin spike, unless it's on something. So I actually make a couple of soups where I drizzle maple syrup into the soup. But the soup is full of plants and flavor and lots of other things. So in that context, the wheat berry salad that I make, one of the key ingredients is pomegranate molasses. And if you leave out the pomegranate molasses, which is just fabulous, which is just sugar, I eat less of the wheat berry salad. And I can eat bowl after bowl of it if I've drizzled some pomegranate molasses on it. So I don't think any of the natural things are any better. They are just sugar. Don't
unless we add fiber to it.
Yeah, right. So here's the, yes, I've seen that. There is sugar with fiber in it now. No, just eat food with fiber, please. Okay, ready for another example? Here we go. How about plant-based meat versus red meat? Oh, wow, Beyond Meat and Impossible Burgers. Oh my gosh. Okay, is alternative plant-based meat better than red meat? Can that be a testable hypothesis? Okay, I'm gonna tell you, because we did this. We published it over the summer. We picked generally healthy adults. Do you agree? Should I pick people with heart disease? I don't know. That's who we picked. You should eat Beyond Meat because they gave us money. So I'm conflicted here. I didn't make any money, but they gave me the money to do the study because the NIH wouldn't. So the Twitter world has accused me of being biased, but actually, I think we did a very unbiased job, but you can get me on that later. What do you think I should pick for a dose? I only get to do one study. You can have red meat or Beyond Meat. Should I pick a hamburger a week? 3 hamburgers a week? A hamburger a day? Only hamburgers. You can only eat hamburgers the whole time. That would be too much. Is there an obvious answer for dose? Oh, what's the obvious? There isn't. We picked 2 servings a day. It ended up being 25% of your calories came from Beyond Meat or red meat, which we bought for you and delivered to your house. For how long? Well, instead of the red meat, so we had the Beyond versus the red meat, and we did it for 8 weeks because we were trying to see if some blood factors would change. And I can usually change blood factors from people who change their diet from A to B in 4 weeks, sometimes 2 weeks. So 8 weeks is really fine. And we actually picked something called trimethylamine oxide, which is sort of a hot emerging new thing to make this sexy in the cardiovascular world. It was discovered by a guy named Stan Hazen at the Cleveland Clinic. Stanford tried to recruit him. I don't know what happened to that. I think he's still at the Cleveland Clinic. Okay, and we called it, I hope you appreciate our acronym. We're very proud of our acronym. Do you like that? Swap meat. I think it's pretty cute. Okay, I got a lot of kudos for our acronym. And we published it last August-ish. And Anthony Cromarco, fabulous postdoc, is the lead author of this, but we have lots of other postdocs in here and team staff members. Maybe some of them are listening. If you guys are listening, I'm acknowledging you. See, if anybody's listening, you were acknowledged. Okay, good. Okay, so we got the meat phase and the plant phase, and it's a crossover study. So everybody was their own control, which makes it possible to have a smaller group. And we used Beyond Meat, and we didn't just use meat. We actually went to a company called Good Eggs in San Francisco, who focus on sustainable, locally grown, regenerative agriculture, organic. We spent a lot of money on the red meat, to be fair. We spent a lot of money on Beyond Meat and a lot of money on this meat. And our study was to get people to cross over, 36 healthy adults, 8 weeks per phase, 2 servings per day. And we drew blood. They picked up food. We collect poop. Justin Sonnenberg is analyzing their stool right now. Did diet assessments, study questionnaires. And here's the TMAO results, which are really unexpected. So we randomized them to which one you got first. I'm actually going to start on this side. The people who got the red meat first, their TMAO levels, which is supposed to be an inflammatory marker and a pro-coagulation, went up, but not in everybody. Can you see in some people it didn't go up? But if you take the mean, it went up over the 8 weeks. And then we switched them to Beyond Meat and it went right back down and stayed down. Okay. So this is kind of what we thought red meat would be bad for TMAO. Okay. We were not expecting this. So the people got Beyond Meat first and their TMAO didn't go up. If anything, it went down a little bit. And then we gave them the red meat and what happened? It didn't go up. Oh, crap. In a crossover study, you want both orders to be the same. This is actually a little bit of a problem that they aren't the same. That means there's a carryover effect. And so actually, Justin Sonnenberg is analyzing this right now. We think, so the generation of this TMAO happens in the microbiome in your gut. And so we did some looking into some other papers, and it looks like when you go on a plant-based diet, you change your microbiome. And the bugs that are in there that make the TMAO go away when you're on a plant-based diet. So we actually, maybe, don't know for sure, protected these people from their TMAO going up by putting them basically on a vegetarian diet first. Wow. So our main result actually was that we lowered TMAO, even though it was only one part of the group, it was still statistically significant overall. They lowered their weight a little bit. They're 1 kilo lower on the plant phase of 8 weeks than the animal phase. That was weird. That's not really statistically, it's not clinically relevant that they lost a couple of pounds, but it's statistically significant because everybody was a little lighter, even though they're getting the same amount of calories. And then their LDL cholesterol went down, which made sense because when they were on the plant meat, they were getting less saturated fat, more fiber. They lost a little bit of weight. Totally makes sense that their LDL went down. So we were able to conclude that the plant meat was better than the red meat at that dose with that kind. Does that mean it works for Impossible? I don't know. We did it with Beyond. Does that mean it works with a new Tyson veggie burger? I don't know. We did it with Beyond. Does that mean it works with 1 dose per day, not 2? I don't know. We did 2 doses. Does that mean it works in people with heart disease? I don't know. We did it with people who are generally healthy who signed up first. I only answered a tiny little question. Right? I didn't answer them all, but there were no data before. And now there are more data.
There is a question, do you recommend individuals getting microbiome assessment?
Oh, not yet. A lot of people can get it done. They don't know what to tell you. We actually assess the participants' microbiome and we give them a profile and we tell you what bacterial species went up or down, but we actually don't know yet what it means. That's why Justin and I are having fun doing a bunch of studies. I actually work with another group in the UK, a group called Zoe, and they have these series of studies, Predict 1 and 2 and 3, and the first Predict study just released in Nature Medicine, its microbiome results, and they propose 15 specific bacteria that are healthy and 15 specific bacteria that are the least healthy. And they didn't necessarily rank them. But the more studies we do, the more we're understanding. I would say don't get it done now. Wait a little bit. We're learning more and more.
There are a couple of more questions. So do you want to answer them now or we wait at the end?
Do you guys want to hear about keto? Why don't I take 2 now and then I'll try to run you through keto.
Sure. About the Beyond Burgers, the question is, they have coconut oil in them. Isn't that bad?
Yep. Coconut oil is saturated fat. So is the saturated fat that's from animal in real burgers. Yep, they're both bad.
Okay.
But what did you have it with and what did you have it instead of, and was it lower?
And the question is, since the dose replacement was 2 per day, does it mean that those subjects were consuming 2 doses, like servings of red meat per day prior to study enrollment as well?
Ah, interesting. You couldn't be vegetarian to join the study. You had to have at least 1 serving a day to qualify to be eligible. And we looked, most people were only having 1 a day. I think if I go back, I think that's why they went up here. These guys were getting about 1 serving a day and we gave them 2. So that's probably part of the explanation why they went up, is because we're actually giving them more meat than they were having before. Okay, let me do keto quickly because I actually have an aspirational diet thing to finish on that I want to tell you guys about. So that I can finish on that positive note, I'm going to do this one kind of quickly. According to my notes, I have 10 minutes, right? We're going till 11, Susan? Okay. All right. So just really quickly, we just finished this study. And here's my personal question. The ketogenic diet is very restrictive. And here's what I know from looking at the American diet. We eat way too much added sugar and way too much refined grain. We don't eat enough vegetables. If you look at the ketogenic diet, it says, got to get rid of added sugar and refined grain. And you should eat non-starchy vegetables. If any of you are up on this, you're supposed to eat non-starchy vegetables and get some carbs from them. So both the Mediterranean and the ketogenic diet, actually, what does this say here? Yeah, are identical in including, avoiding these carbohydrate sources, Mediterranean. And here's what I'm frustrated about is the keto diet says no beans or legumes, no fruits, and no grains, even if they're whole intact grains. Whereas the Mediterranean diet would say, yes, eat lots of legumes and fruits and whole intact grains. So my personal question here is, if you did this, if you just got rid of the added sugars and refined grains, would you really get an additional benefit from avoiding these, which the whole public health community thinks is healthy? Okay, I question that. I think our focus should be here, not here. So the 2 diets are the same in this, and they're different in that. Okay, for the sake of time, I'm going to have to skip this. The hypothesis is one will be better than the other. We focused on the well-formulated ketogenic diet. So again, we tried to make a really healthy ketogenic diet, to be fair. And then we tried to have a really healthy Mediterranean diet, whereas the absence of added sugar and refined grain isn't technically part of the definition. You saw the definition before in one of the other studies I showed you. So he said it's all the same things as the other studies, including olive oil, plus you should get rid of added sugars and refined grains. So we're going to focus on glycosylated hemoglobin, but we are measuring tons of other things. So I'm going really fast so I can finish on my aspirational note. It was once again a crossover diet. So 20 on each arm. This is actually 10 on each arm with type 2 diabetes and 10 with prediabetes for 20 total, 40 altogether. Collected same kind of things, blood, poop, diet assessment, study questionnaires. And a twist to this was to make it as rigorous as we could. We actually, for 4 weeks, delivered food for the first 4 of 12 weeks. And then they switched to the other diet. And we delivered food so they would be instantly adherent. And then they had to be dietitian supported and learn on their own. So it was generalizable. And we have a follow-up time point. I want to see after the study's all over what they choose to eat 12 weeks later after the study's over. That's actually one of my big questions. Is it sustainable? Okay, so this fabulous company that delivered boxes of food. And here's a participant who took a picture of all their food for the week. And that's the Mediterranean in the fridge for the week. Okay, sorry, going fast. I have to skip this. This is all my cool diet data. I'm just going to tell you they did a pretty good job. But the bottom line is when you're getting delivered, you're more adherent than when you're on your own, as everybody would expect, right? And so we came up with a score. I actually don't have the final results now. I just want to show you something really practical. We came up with a score of how well you were doing on keto and a score for how well you were doing on med. And my brilliant postdoc, Matthew Landry, said, okay, I know they actually aren't assigned to a diet at baseline, but how would they have scored on the keto diet at baseline? And they would have scored really poorly. 10 is best, 0 is worst. And then when we delivered food, wow, they did really good, but they weren't perfect, even though we delivered it. What did that person do? Did they throw all the food away that we gave them? Damn it. Oh, we should probably study rats because rats only eat what you give them. Okay, but these are humans. So this is the variability that we got. And then this is what they were when they're providing their own food, their adherence went down, even though we showed them exactly what to do. We delivered it. It was harder to do on their own. And then at the end, they didn't really stick with any of these. They're pretty low for keto when it's all over. Does that make sense? And we'll go over to med. They actually scored higher at baseline just because normal people's diets are more Mediterranean-like than keto diet. This is super restrictive. So even if you're not trying to be Mediterranean, you scored higher on Mediterranean baseline. Here's where we're intervening. And so they did better, right? But not everybody did the same, and they got worse. And then they weren't quite so much on it. So for internal validity of the study, this is pretty cool because according to our score, when we were feeding them the two diets, delivering the food, they were both pretty similar in adherence, 7.5 out of 10. And then when they were providing it for themselves, they were pretty adherent, only like 6 out of 10, but still way better than baseline. Okay, I'm just gonna skip. Cool, I'm just gonna show you that we don't have the glycosylated hemoglobin. I just gotta show you the weight change data, okay? So here's a teaser. In the very beginning, keto lost a lot of weight really fast when we were delivering the food, and the Mediterranean diet lost some weight. Look what happens when they provide food on their own. Their weight comes flying back on because restrictive diets are difficult to maintain. So when they switched over to the other diet, these guys lost a little more weight when they got to keto. And these guys, when they switched to med, their weight totally stabilized. And this is going to be a really fun time point because out here, after they've done both, regardless of which one they did second, they ended up with the same amount of weight. I actually think their glycosylated hemoglobin is going to be really similar right here. And how did they do after 12 weeks of follow-up? Bless their hearts, they maintained that weight loss. But if you do both diets, no matter which one you end up on, you end up with the same weight. Okay, that's my teaser. Okay, I have to skip all this. Okay, now I'm going to try to inspire. This is sort of inspirational. I'll try to do this quickly. This is a bad question. Is the ketogenic diet healthy? Compared to what? Okay, is it healthier than the usual diet? Well, the usual diet in the US sucks, so that's not a good comparison. How about versus really low-fat vegan diets? Well, that's one possibility, but I actually was more interested in sort of an optional low carb diet, a Mediterranean low carb that was high in olive oil, but also high in legumes and beans and whole grains. For what? Oh yeah, right, you have to say for what. So we picked glycosylated hemoglobin, and you have to say for who. Oh yeah, we said this group would be more relevant. And then we could have done a 2-week metabolic ward study like Kevin Hall does, but I like more generalizable than that. So we did maybe a 4-week feeding study. We did a 4-week feeding study and 8 weeks of self-provided food. So I hope you appreciate my interest in balancing rigor and generalizability and even having that sustainable thing out here. So we're trying to answer a couple of questions at once for keto and med. Stay tuned. I can't tell you what happened. I'm actually still blinded to the glycosylated hemoglobin results, but I think I'm going to know this Thursday or next Thursday. Drum roll. Okay, skip this. Okay, let's leave you aspirationally. I think most people in my field actually agree on most of this stuff. I think when it gets to grains, we have gluten intolerant people, and poultry, we have Kentucky Fried Chicken versus, pasture raised eggs and chicken. And okay, whole intact grains is what I'd focus on. Dairy and red meat are the most controversial. I know in the newspapers it looks like we disagree a lot, but I really think even the ketogenic people and the low-fat vegan people agree on all these things. These days, I try to focus on what everybody agrees on more than what they disagree on. And I really think there's considerable agreement, even though in the newspapers and on Twitter, it looks like they disagree. That is both vegan and keto. That is vegetarian and might be keto except for the orange in the back. This isn't keto because it's on a bed of sprouted beans, but wow, that is a really super healthy dish. So a lot of this has to be instead of what, right? A veggie omelet versus steel cut oats versus Pop-Tarts. I don't know, I could argue back and forth between the veggie omelet and the steel cut oats, but they're both better than Pop-Tarts. Those are both whole food, plant-based. The Pop-Tarts are not. And lately, I've been encouraging students to think about things other than nutrients to change their diet. I teach a class called Food and Society, and I find that when students get involved in social justice issues and other things like that, the environment, they actually make pretty good changes to their diet, and it's got to taste good. So I now collaborate with the Culinary Institute of America on unapologetically delicious food. So here it is, my aspirational diet. Yes, it should be healthy and nutritious. I have a PhD in nutrition science. It should taste good. If it doesn't do that, people won't switch. And I really think we could put environmental sustainability and social justice into it for an aspirational diet. That is my goal, pushing all of those domains. And so it turns out that this talk, after Susan assigned it to me, I realized that a bunch of my slides had to do with an invited lecture I gave for American Heart Association's Epi and Lifestyle Group. And so you will now understand my concluding slide from that talk that I gave. I think we have agreement. I think we have to embrace variability. Not everybody responds the same way. I think we have to understand the balance and trade-off between rigor and generalizability and know if we're talking about nutrients or foods or patterns, and keep in mind instead of what. I hope everybody will eat responsibly, because our food choices have huge effects on other things. And what I really want y'all to leave this talk with is that you should eat joyfully and deliciously. Stop taking that away from nutrition. We really need to get back to that so that people will enjoy what they're eating. And if you want more and you have $2,300, okay, shameless self-promotion, use your STAP funds. I have an online nutrition course that's way longer than this 2-hour thing. Susan, thank you for inviting me. I hope you guys like this. I'm happy to stay for more questions, but you guys put in your 2 hours and I think you get your credit.
Yes, wonderful. Thank you so much, Dr. Gardner. It was really wonderful information and some of it very eye-opening. Thank you so much. And for the participant, we will send out an evaluation. So to get credit, you will need to complete the evaluation. And if you want credit, you need to put your name, a full name in the evaluation. You need to complete the evaluation within 30 days. And again, for those of you who can stay beyond the hour, we have a few questions. First, Dr. Gardner, can you stay or do you have to go?
Just for a couple of minutes. I could. If there's some burning questions, I'd like to try to get to them.
Okay, first people are asking for your wheat berry salad recipe. If you could share that later on.
Okay.
You can email it to me and I will share it with the participants.
I will.
And then also a quick question, is coconut, avocado, tofu, like full, are they bad overall because they are, you know, they have cholesterol? So that's one question.
No, so no plants have cholesterol. So that part was wrong. No plants have cholesterol. Coconut has saturated fat.
Okay.
Avocado has unsaturated fat. Avocado is a huge win anytime. Coconut, shakier. Just have a little bit of coconut oil.
Okay. And there are other questions. It seems that a lot of these available dietary studies are in white maize, and how generalizable that can be across genders and races.
Yep, it's embarrassing. Yep, we're getting better at cultural diversity and gender diversity. Actually, I have to say, most of the people who sign up for my studies are women, because women care more about their health. So when you look, I actually have to try really hard to get men in diet studies. So keep looking. We really do need to work on diversity though. So we are trying to get more culturally diverse folks signing up for our studies too.
Okay, one more question. Are there certain diets that may be more beneficial for people with hypothyroidism?
That's a bad question for me. I can't answer that one. I really don't know much about hypothyroidism.
Then we skip that. Another question, taking multivitamins may not add any benefit because it may create imbalance when a person's diet is reasonably good, as it may complete with minerals already in. Please comment.
Yeah, so there have been randomized trials on multivitamins, multiminerals. They don't really hurt and they don't really help. They tend to get taken by people who already had an adequate diet with adequate resources and variety. I would say if you are a medical intern or resident and you're working huge shifts and you can't eat right, if you are a person who doesn't have access to a good variety of diet, if you have a picky kid, who's young, there are places for a multivitamin and multimineral. So if there is a chance that you're just not eating a good balanced diet, there's little to lose and something to gain. For the general population, I don't think it's worth it. If you are going to get something like a Centrum Silver that has 100% of the recommended daily allowance. If you look at some of the supplements, they have 1,000% of the recommended daily allowance. Why would you want 1,000%? Just get 100%. So choose one that has 100% of the nutrients, not more. And maybe one more, Susan.
Yeah, one last question came in actually. High blood pressure is becoming increasingly common. Any advice for lowering high blood pressure through nutrition? Low red meat and salt, increase veggie, what is?
Yeah, so look at the DASH diet. There's a website. There's been some fabulous DASH study diets, and they do two things. They lower sodium and they increase plants. And the plants have a lot of potassium. And so the potassium and sodium balance is pretty important. So there's one of the few areas where for hypertension, we have randomized controlled trials because again, blood pressure is a risk factor that you can change in a few weeks. They've done them. Look up the DASH website. It's Dietary Approaches to Stop Hypertension, DASH. Susan, thank you. And all your
listeners. Thank you. We really appreciate it. And thank you so much for staying a few minutes beyond the hour. Have a wonderful day. Thank you. Thanks, everyone.
Thanks, everybody. Wish I could have seen you.
Bye.